Let us summarize this. What are mutations? Mutations are accidental changes in the genes, in the DNA. They alter the existing order. They may have no effect, neutral mutations. We know many examples. They may spoil the gene, cause it to malfunction or fail to function. We know many examples with field mutations. Improvements are postulated but not documented. And it is suggested that changes that bring resistance to man-made chemicals, such as antibiotics, pesticides, herbicides, that these changes are examples of positive mutations. In fact, they defend the existing order and functions. Most of such changes are achieved through recombinations of existing genes or from immunological adaptation. There are very few examples of resistance achieved by mutations. Next slide. One such example is the mutation in a plant called Amaranthus hybridus, which achieved the resistance to a man-made chemical, atrazine. Atrazine was specifically developed to eliminate Amaranthus hybridus from the population. This is a weed. It's a weed we don't want. We use the herbicide to kill off the weed. Nothing else but the atrazine. Now, this was specifically developed for this particular plant. And it operates in such a manner that atrazine, this chemical, attaches to a protein, which is symbolized by QB, which is present in the plant. It is coded by gene PSBA. And this atrazine QB complex prevents photosynthesis. So the plant is unable to live and it dies. Atrazine attaches to a segment of this protein. And in that particular segment, in one place, there is the amino acid serine. In DNA, serine is coded by the 3 nucleotides AGT. This triplet is found in this gene PSBA. Now, one nucleotide change from A to G, this change, this is in the 682 position within the gene and so on. And this gives us, instead of AGT, a GGT triplet, which is coded for a different amino acid, namely glycine. And atrazine does not attach to the variant with glycine. Thus, the mutation of one nucleotide from A to G gives resistance to atrazine to this plant, Amaranthus hybridus. Now, the evolutionary conclusion is this, that mutation of one nucleotide from A to G gave resistance to atrazine. This resistance is considered a new function, and this example is advertised as evolution in action. Now, I criticize this conclusion. The change defends the functionality of QB, its role in photosynthesis. No new function is formed. The function of QB is defended by this change. The change is at best a neutral mutation. It maintains the function that this protein is playing in photosynthesis. The protein QB with the variant with glycine, that is in the DNA with the triplet GGT, is less adapted to life, because when atrazine is eliminated from the population, which is not used, the population returns back to the natural variant with serine. Thus, the mutation is in fact slightly negative. In nature, it is not preferred.